All right, in this video lecture, we're finally going to move on to the B lymphocytes or B cells, and as we'll see, when these cells get activated, they go on to produce antibodies, and antibodies are very critical weapons that your immune system uses to help clear out very specific infectious microorganisms. All right, so we have already discussed earlier that B cells, like T cells, have receptors on their surfaces. However, in the last video clip, we talked quite a bit about how your helper T cells see antigens that are antigen fragments that are presented to them inside MHC class II proteins, and those MHC class II proteins are present on phagocytic cells like macrophages and dendritic cells. And then we saw how the cytotoxic T cells, they have receptors that bind to antigen fragments that are cupped inside MHC class I proteins, and MHC class I proteins are present on essentially any cell in your body that has a nucleus. And that's a great thing because almost any cell in your body could be the target for some sort of virus or another intracellular pathogen. And nicely for us, when one of your cells is invaded by a microorganism, that cell is going to display little antigen pieces on its surface to try to attract in these cytotoxic T cells to torpedo them. So the cell is going to wind up getting killed to help limit the spread of the microorganism. So that's all very important. But what's different about the B cells, they have receptors on their surfaces as well, but they bind to soluble antigens. Okay, and so that could include a molecule that's foreign to the body, a molecule of some sort of a pathogen. Remember those exotoxins we learned about back in Chapter 11, bacterial exotoxins, or molecules that are on the surface of a microbe, on the surface of a bacterial cell, on a yeast, on a virus, whatever. So B cells don't bind to presented antigens. It binds to the antigens themselves, essentially. All right, and like the T cells, individual B cell clones, a single B cell is going to be covered with these B cell receptors, and all of those receptors are going to bind to the same antigen. So that's important as well. So you've got this B cell diversity, you've got B cells with receptors that can attach to, almost any antigen that we get exposed to. Then as we're going to see, so things are different with B cells though. When a B cell attaches to its antigen that it's programmed to bind, it's actually going to do endocytosis, or essentially like phagocytosis as well. It's going to bring in that antigen. It's going to digest it, just like we've seen with phagocytic cells. And the B cell is going to wind up presenting fragments of that antigen on its surface inside MHC class II proteins. And why is that happening? So where did we see that before? So we just mentioned that MHC class II proteins are present on cells that show antigens to helper T cells to try to stimulate those. And so we'll see why this is for B cells shortly. So let's kind of take a look at our, just a reminder about what the surface of a B cell looks like. So obviously we've got a cell membrane here, and notice right here, you've got many copies of MHC class II proteins on the surface, and then the Y-shaped molecules that we're seeing there, right here and elsewhere, those are your B cell receptors. And that Y shape reminds us of what antibodies look like. So a stimulated B cell, a fully activated B cell, is going to go on to produce and secrete lots of antibodies. But on the surface of the B cell, it's actually covered with antibodies that function as B cell receptors. And so as I just mentioned, let's say we've got a virus particle here. So that virus particle is going to have molecules on its surface that are antigens, functioning as antigens. And somewhere there are going to be B cells located with receptors that will attach to that particular antigen. So you might have another copy of the same virus over here, and maybe another copy of the same virus over here. And kind of just like the T cells, the B cells are hanging out in your lymph nodes and your spleen and your tonsils and so forth, because those are great places to intercept something like a virus that's invading the body, or bacteria or yeast or protozoans, whatever they may be. As those types of things drain in from an inflamed, infected tissue area, they're going to bounce around inside a lymph node and hopefully encounter a B cell that has receptors on its surface that can actually attach to it. So that's going to be very critical. So it's pretty amazing when you think about this, that all this actually works. When you think about all the different types of microorganisms and other foreign things that we get exposed to, it's pretty amazing that those things inevitably are going to encounter B cells and T cells that they can react with. All right, so B cells receive help from helper T cells. We talked a little bit about that in the last video clip. And really, I should say here, many or most B cells, not all of them. So there are some B cells that can get activated without helper T cell assistance. And that occurs with something called T-independent antigens, and you'll read about that in your textbook. And what that means is there are some antigens that are capable of stimulating B cells, getting them activated without any assistance from a helper T cell. But we're going to really focus in on the B cells that do need help to become fully activated. So let's take a look at what's going on here in this diagram. So as you guys can see, we've got a microbe here, looks like kind of a generic virus, and that's draining into a lymph node, or maybe it's entered into a tonsil, or if that's a virus in the bloodstream, maybe it is encountering B cells inside the spleen somewhere, or this could be down in the intestinal tract, and this B cell is in a little patch of the gut-associated lymphoid tissue. Anywhere, those are the places where B cells tend to encounter these microorganisms. And so that microbe is going to wind up, if the receptors on the B cell attach to one of its antigen molecules, those two will come together. And then what's going to happen is the B cell here is actually going to do phagocytosis. It's going to bring in the, or we could call it endocytosis at this point. It's going to be endocytosed. You're bringing in the microorganism, and same story like we've seen with your phagocytic cells. That microorganism is going to get digested, broken apart by the digestive enzymes from lysosomes. And then the B cell, remember we said B cells have class 2 MHC proteins on the surface. And so that's what we're seeing right here. So that B cell is going to display on its surface little pieces of the antigens of that microbe. And then notice what's going on over here. We've got a helper T cell. So remember, helper T cells attach to antigen fragments that are being presented in class 2 MHC molecules. And that's what we're seeing here if you kind of zoom in on this figure from your textbook. The B cell, there's an antigen piece presented in an MHC class 2. And then over here, the helper T cell has a receptor that is attaching to that specific antigen piece. And notice right there you've got the CD4 that is helping to bridge the two together. All right, once that happens, once those two come together, notice that the helper T cell is going to secrete B cell growth factors. So it is going to secrete proteins that are signaling molecules that are going to tell that B cell to become activated. So the B cell is going to begin to divide over and over and over again. Now, interestingly, when you think, all right, now why does this have to happen? Why do we have to go through all this rigmarole? Why doesn't the B cell, once it attaches to its microbe, why doesn't it just automatically begin to divide over and over again and become stimulated? This is very likely a safety mechanism. So we've talked a little bit about autoimmunity, you know, how we don't want B cells that react to our own antigens. Remember we mentioned clonal deletion. So any B cells that are generated that can react to your own antigen molecules, any self molecules, those are supposed to die. Those B cells get tested before they leave the bone marrow, and they're supposed to die. But as you can imagine, sometimes that's not going to work. So these B cells may wind up being released into your circulation. And so imagine if that happened, it could be pretty easy for a B cell to begin to react to one of your own self molecules. And if that happens, then you're going to start making antibody weapons against your own self molecules. And we don't want that to happen, because if it does, then you're going to have an autoimmune disease. So this is kind of a safety mechanism. It's like two switches have to be flipped before a B cell is going to become activated and start cranking out lots of antibodies. So switch number one is right here. The B cell has to see the antigen that it is programmed to attach to and respond to. And then also over here, you have to have a helper T cell that has also seen that same antigen. So if you remember from the last little video clip, we talked about how helper T cells get activated. So in order for these two to come together, that helper T cell has already seen pieces of the antigen and had that presented to it by a macrophage or a dendritic cell. It has already previously been stimulated. So that's switch number two. So you have both a B cell that saw an antigen and a helper T cell that saw an antigen. So imagine if you're thinking about self molecules. Yes, it might be possible for a B cell to escape that reacts to one of your own molecules when it wasn't supposed to. But it's unlikely that you would have both a B cell and a T cell that escaped that react to the same self antigen, the same self molecule. So what this does, it makes it less likely that you are going to have inappropriate B cell stimulation to your own self molecules. It's basically a safety mechanism that takes place. Oops, hang on. All right, so what happens after this B cell gets activated? So this is going to lead to something called humoral immunity. If you see that term, that is referring to antibody immunity. Humor is an older term for body fluids. Antibodies are proteins. They're protein molecules. They're going to be dissolved in your body fluids or body humors. That's an older term. And so that term, and it's kind of older terminology, but you may still hear it used potentially in future classes. So if you hear anybody talk about humoral immunity, that means the antibodies that have been generated against some foreign invader to the body. And remember, we said T cell immunity is sometimes referred to as cell-mediated immunity. So just be sure you're familiar with that terminology, because it does come up occasionally, and probably will for you in some of your future classes. All right, so let's say we're starting here. We've got a B cell that has become activated. And again, not all B cells have to have that co-stimulation from a helper T cell. There are some that respond to what are called T-independent antigens. So just the antigen by itself will activate the B cell. But most of them do. Most of them, you got to have that connection, that kissing going on between the B cell and the helper T cell before it's going to become fully activated. So this diagram doesn't really show it, but when that B cell gets activated, it is going to divide over and over and over again. So it's going to undergo clonal expansion. And so you're not going to have just 3 copies like you see here. There are going to be many, 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 many clones of the original. And some of those B cells will then go on to become what are called regulatory B cells. And we're not going to worry about that. Again, that gets into, the immune system is very complex. You want to have a reaction, but then at some point you got to dial down that reaction. And that's where these regulatory B cells and T cells get involved. If you ever take an immunology class in the future, like a whole semester on the immune system, then you can learn more about how things are regulated. And my understanding is that that's still not all that well understood. The immune system is very complex, and many of those details are continuing to be worked out. So we're going to kind of ignore the regulatory B cells. Most of those clones become plasma cells. So you see these oval-shaped cells over here. So the activated B cell clones, most of them take on this kind of oval shape, and they basically become antibody factories. All right, so they start secreting tons and tons of antibodies. So these little things over here that look like snowflakes, those are the earliest antibodies that are made by plasma cells. Have a snowflake type appearance, and we're going to talk about the different types of antibodies that plasma cells can secrete shortly. So it's secreting these antibodies. If this is happening in a lymph node or wherever, those antibodies are proteins, so they're going to get into the body fluids. They're going to travel through the lymph. They're going to be dumped back into the blood. They're going to float through the blood. If you have an area of inflammation where an infection has taken place, the antibodies, well, many antibody types can get out of the blood and into the tissues to help fight infection. That's what happens with the majority of them. However, notice over here, we also have memory B cells. So we talked about how when a T cell gets activated, there's a certain subset of the clones that really don't do anything right now. They just go on to become long-lived memory T cells. And same thing with B cells. There's a subset of the clones that have been activated, become these long-lived memory B cells that hang out in your lymphatic tissues, and they're basically waiting around to see if you ever get exposed to this same antigen again, because if you do, they're going to respond really, really quickly. You're not going to have to go through as much of this rigmarole that we've just been talking about. They're going to be able to respond super fast and start cranking out these antibodies really, really, really quickly the next time you get exposed to the same pathogen. All right, so that is some introduction on how we get B cells stimulated. And again, you got to kind of work with the terminology here. So the cells that actually secrete the antibodies are called plasma cells. So activated B cells, most of those become plasma cells, and plasma cells are antibody-producing machines. That's their job, is to crank out lots and lots of antibodies. Oh, and one thing I didn't really point out over here. So up here at the beginning of the story, this was our little generic microbe that attached to the B cell receptor originally. The plasma cells over here that are generated from that activated B cell secrete these antibodies. Those antibodies bind to the same exact antigen. So here's our little generic virus that we have here, and the antibodies are attaching to the same molecule that attached up here to the B cell receptor originally. So you're making very specific weapons that are going to attach only to the microorganism that stimulated that B cell originally in the first place. So in the next video lecture, we're going to talk more about antibodies, what they are, the different types of antibodies, and the special locations and jobs that they have, because these are an extremely important set of weaponry for the full function, the full power of your immune system to be realized. So we'll learn more about antibodies when we get into the 5th video lecture for Chapter 13.